Hi, this is Teresa Tufts. Sorry I couldn't be there in person today, or even in person on screen for that matter. We've had so many technical difficulties, so this will have to do. Um, Before I get started, there are numerous groups that I have to thank that have made my research possible. So thank you, big thank you to all these groups. So the diversity we see in our world today is the result of adaptation. We all know this. Organisms that you see out there, they're very closely associated with their environment. They're adapted to the abiotic and biotic factors that they're facing every day, um, and they're locally adapted phenotypes. So these locally adapted phenotypes are the result of uh, their genes and their environment. So their phenotype um, is what comes out of that interaction, and this process is shaped by natural selection, but it can also be driven by phenotypic plasticity. What happens though when one of those elements changes? So the reality we're facing today is climate change and changes that are happening at a rate that might be too fast for organisms to keep up with. So in this dynamic, we have a phenotype that's now mismatched with the environment. So the question that we want to figure out is, how will species respond to this change, and will they be able to respond? In my research, I look at lake environments, and it turns out animals that live in lakes might be particularly at risk. This study shows the difference in lake temperatures between 1985 and 2009. So a red dot indicates that that lake has warmed, and blue indicates that that lake has cooled during that time period. So if you look at this, you can kind of see that most of those dots are red, and in some cases, those lakes warmed up to twice as fast as the air temperatures warmed during that same period. This is a problem for ectotherm species that are living in those lakes. Ectotherms are organisms that inherently rely on the temperature of their environment to undergo any of their physiological processes. So things like fish and frogs and lizards, those are ectotherms. So their physiological functions are, they function on this curve, and it's bound by a minimum and a maximum temperature. So outside of those boundaries, that organism loses all function, and it ultimately leads to death. And you can imagine that because these species are so closely associated with their environment, when that environment changes, it's going to really influence how their biology behaves. Will they even be able to survive? That's the question. And at this point, if they're going to survive, they're either going to have to change their behavior, they're going to have to adapt, or they're going to have to use some sort of plastic response. So here's an example of an ectotherm adapting with its behavior. So it's choosing to sun itself where it's warmer than in the surrounding habitat. And this is pretty common for terrestrial ectotherms. The terrestrial environment is more complex, and there are more microclimates that these animals can use. In comparison, aquatic habitats are more homogeneous. They have less complexity, and the animals have less of these options in microclimates. And that's particularly true in freshwater systems compared to marine systems. Fish are looking a little bit like a mix between these two graphs, but more so towards the one on the right, where their optimum is getting much closer to that maximum. My study looks at the Ninespine Stickleback. It is a cool little fish. It's super hardy. It's super charismatic. Um, if you ever go out to a lake or a stream and find them, take a closer look at them because they don't look like much, but they're actually super cool. And you can learn so much about them and their habitats. So the Ninespine is the perfect organism for me because it's distributed across the northern hemisphere and throughout a range of different environments. And that allows me to test how populations adapted to different environments will respond to changes in temperature. So I'm using representative populations from the northern and southern part of their range. I look at populations in northern Alberta and also from Baffin Island, Nunavut. My research questions are, uh, they're in three parts. So the first one is, do representative southern and northern populations exhibit variation in thermal tolerance range? So basically, do populations that are adapted to totally different habitats respond differently to temperature change? And do they even have different thermal phenotypes? So just characterizing what their phenotype is in those different environments. The second part is, are these traits genetic? Is there a genetic basis to thermal tolerance? Or, my last question, is there plasticity? And if there is plasticity, is it constrained at higher temperatures? So if you remember that graph with the colors and the horizontal black line and the diagonal black line, where in that spectrum do the Ninespine lie? Are they able to compensate for temperature change, or are they sort of stuck? So I've collected fish from 2 sites in Nunavut. Um, these lakes have low productivity. There's also quite low predation. There's not many species that live here. In fact, there are only 2 species, the Arctic char and the stickleback. My sites in northern Alberta are here. These are my representative southern populations. These lakes are much more productive. There are many fish species that live here, so the predation rate on these stickleback is much higher. So those fish have come back to the lab at the University of Calgary, and they're under common garden conditions. So they're at 17 degrees, both Alberta and Nunavut populations, and they've been acclimatizing to that for a couple months. And recently I've done my trials, so to test what those physiological limits are, their thermal tolerance limits, I use this setup where for the minimum I cool a water bath. I cool the water in this holding tank with dry ice and with water chillers. That gets pumped into a water bath where the beakers with the fish are, and this gradually cools the water in the beaker down to the point where the fish can no longer stand that temperature. So they sort of start to roll over on their side. Um, at that point, I take them out and they recover. So don't worry, this is non-lethal. The fish live. And similarly for the upper end of that spectrum, their maximum tolerance, I heat the water using aquarium heaters, and the same idea, I heat it to the point where the fish can no longer stand that temperature. Here's just a close-up of the beaker with the fish in it. I also make crosses with the eggs of a mature female, which is on the left, and the testes of a mature male, which is on the right. And I make across, so the babies will hatch in the lab, and that allows me to compare the parent population to this baby population without the confounding variable of their environment and where they grew up, and I can test if the traits I'm observing are genetically based. I have some preliminary results. So this is for the maximum temperature tolerance of the adult fish. So the population in the middle is the Alberta population, and it happens to be a little bit higher than the Nunavut populations, but not by much. So it's pretty remarkable that these Nunavut populations can withstand up to 31, 32 degrees. It's really neat. And on the other end of the spectrum, the minimum temperature they can withstand, you can see their means are all pretty similar, but the population in Nunavut is slightly lower than the others. Okay, so how do they respond though to prolonged warming? That's what scientists want to know. We want to know what's going to happen in the coming years when those lakes heat up. Are these fish going to roll over and die, or are they going to be able to adapt and adjust their thermal tolerance? So to test that, I have an experiment running in the lab now with 3 different treatments. So one is a control where I haven't changed the temperature, and then the other 2 are elevated temperatures, and I do that using these aquarium heaters. And so they'll be exposed to these conditions for 3 months, and after 3 months I'll test those thermal tolerance limits again, and I'll see if there's a difference in response. So if the treatment groups have a different thermal tolerance range than the control group, it'll suggest that fish are able to plastically accommodate that temperature change. So they're not stuck because of their genetics, but they can actually adjust based on what environment they're in. So if the fish do not show difference between the control and the treatment, like I was saying, or no difference between the parent and the baby, it might suggest that these traits are genetic and they might actually be stuck. There might actually be constraints to their ability to adapt, and they might actually be in trouble. But on the other hand, if the treatment and control are different, it'll suggest that they can adapt, or they can use plasticity to persist, and that's a good thing. So I hope for my study to tell us a little bit about fish in general and use my results to make predictions about these other fish that are maybe more important to us economically and culturally, like the Arctic char. Of course, with anything, my study has limitations. The process of adaptation is very complex. There's a lot going on, and there's lots of feedback, and my study is just going to scratch the surface. So it'll tell us a lot, but it's inherently limited. That's all I have. If you have any questions for me or comments, here's my email. I'd very much welcome your feedback or questions, or if you just want to know more about what I'm doing, shoot me an email and I'd be happy to hear from you. Thank you so much.